All right, good afternoon, everyone. This is going to be chapter 6. It's on metabolism, energy, and enzymes. This is going to be a precursor chapter to our probably two most difficult chapters on cellular respiration and photosynthesis. Basically, we're going to kind of take a step back and we're going to look at enzymes, which are types of proteins that are like the workers of your cell. So, in this chapter, we're going to look at energy first, and then we're going to work our way to enzymes and then factors that affect enzymes. All right, let's get into it. So there's a lot here in the beginning where I don't have you, take notes because you should know a lot of this stuff. Energy, this is just the ability to do work. We talk about kinetic energy being, energy of motion, and potential energy having the potential to do or stored energy. But yeah, basically, we're going to be looking at in the next two chapters how chemical energy is stored in food and how do we break that energy down or how do we transfer that energy, which is going to lead me into my next topic here on the laws of thermodynamics. So the first law of thermodynamics, also called the law of conservation of energy, states that energy cannot be created or destroyed. It has to be transferred from one to another. So again, you can't just produce energy. You can't just create energy. You have to transfer it. So talking about like food, we can get energy from food, but it's not like we're producing that energy. We have to transfer that energy from food into our ATP molecules. So that's the first law of thermodynamics. Second law of thermodynamics states that energy cannot be changed from one form to another without some of it being lost. We're going to talk about why that's important, especially evolutionarily, but just how when you transfer energy, you're not going to get 100% of that conversion, and you're going to lose some. Usually due to heat, but other factors as well. All right, so these are like that's the main kind of topics about um, uh, energy. We talked a little bit about this, how about how we get energy from our food and we transfer it to our ATP. Where I always ask them in class, where do we get this energy, or where does the food come from? It comes from plants. Where do the plants get the energy from? The answer there is the sun. So essentially, we're transferring that energy we get from the sun into the plants, into the food, and then into us, and then we use that as potential and kinetic energy. So again, this whole first section is about energy. I know we haven't taken any notes yet, but we're going to get right into it. There we go. Metabolism is the sum of all the chemical reactions that occur in the cell. I have a big chart, and I have to find it. I don't know where it is in my classroom. It's a big chart that shows all of the metabolic pathways that occur in your cells, and it's just really cool and interesting. And it can be overwhelming at first, but I love it now. It's almost like art to me. But we're going to talk more about reactants and products in the next couple chapters about what goes into each step and then what comes out, or what is used up and what is produced. All right. Let's go on to the next slide. Okay. Exergonic and endergonic reactions. Do understand, I am giving you a biological explanation of these reactions. In chemistry, you might hear some things different. I'm not going to. It's almost like a funny debate where with breaking and forming bonds, but in this class, exergonic reactions that release energy. Think exergonic exiting. So cellular respiration, I classify as an exergonic reaction because you are breaking the bonds and you're producing energy in some way. Endergonic reactions, it requires an input of energy. This is like photosynthesis. This is the putting in of energy, putting in of the sunlight energy to form carbohydrates. Now, Gibbs free energy, this is going to be more for the CHS students, but Gibbs free energy is called delta G. Delta G is the change, or Gibbs free energy is the change in free energy, and this is basically just the usable energy to do work. Basically, all you need to really know is if you have a negative delta G, energy is being released. If you have a positive delta G, energy is being consumed. And we'll talk a little bit more about that in the next slide. There we go. So we have this delta G, Gibbs free energy. We have the reactants, we have the products. Energy here is going to be released. So in an exergonic reaction, we start with more energy than we end with because energy is being released. In an endergonic reaction, you can see our reactants and products. We have to put in energy. Energy is going to be consumed in this, okay? So when talking about these two different processes, I like to talk about the heat. If you have a fire and it's burning, you're releasing energy. It feels warm. Endergonic reactions, think about those cold packs when you break them. Energy is going in, so it feels cold. So going back to these slides here, again, we're releasing energy. The reactants had more energy than the products. That's like the heat from the fire. When you have your endergonic reactions, you're putting in energy that's feeling cold because energy is going into that system. All right, talking about ATP, the same thing we talked about with the energy in, energy out. ATP, you break down a phosphate group releasing energy. negative delta G, then you have positive delta G because you're putting in energy to form that high energy bond up. So again, ATP, 3 phosphate groups, ADP, 2 phosphate groups. We get energy from ATP here. You can see the phosphate group leaving. So we're getting energy. It's powering our systems. And then you have to put in energy to form that. That's like the cellular, that's like the photosynthesis. Now, looking at this process, when you break down ATP, energy is being released. Chemists, just, I get it. It's not what you think, but okay. When you consume energy, it's when you form ATP from bonding a phosphate group onto ADP. That means that there is a negative delta G for ATP because energy is being released, and you have a positive delta G because you have to energy is being consumed. This is an exergonic reaction. Again, you're releasing energy. Endergonic, you have to put energy in. And then, which has more energy? Obviously, the reactants has more energy because you're going to lose some of that energy in the form of heat. And which has more energy? Obviously, the products, because you're putting energy, it's being consumed. I would definitely know this chart for the CHS students. All right, hello everyone. This is going to be 6.2 on enzymes. Enzymes are protein molecules that function to speed up chemical reactions. And there are two main things that they do is they either break things down or they like split bonds apart, or they build things up or they join things together. So this enzyme here in purple, you can see is breaking down this disaccharide. So we can see that this disaccharide was originally 2 monosaccharides bonded together. We can see it was sucrose. What this enzyme is going to do is it's going to break these monosaccharides apart. Now, if you remember, this is a process called hydrolysis because we're using water to break things down. But basically, this specific enzyme is going to break down a disaccharide into 2 monosaccharides. So again, proteins function to speed up reactions, and they either break things down or build things together or join things together. Now, a couple different things about enzymes. Like I said, they either join things together or they break things apart. They are reusable. And they usually end in ase. So they usually end in that suffix of ase. So like lactase breaks down lactose. But yeah, here we go. Those are enzymes. Now, a couple different things to remember about enzymes. There's a lot of different terminology here that you should know. So again, enzymes, this is the actual protein that's I always make the analogy like they're like scissors or they're like a stapler, or they're going to cut things apart, join things together. But what they actually work on is called the substrate. And the substrate is the reactant in this reaction. The active site is the location where the enzyme is going to come in direct contact with that substrate. So like in a pair of scissors, it's where the scissors are cutting the paper, or with the stapler, it's where the stapler is connecting the paper together. The enzyme substrate complex is the intermediate formed when the substrate attaches to the active site. And then the product is what is formed afterwards. And I like showing students this kind of like equation. So we have our enzyme plus our substrate. It forms our enzyme substrate complex. And then our product is formed from our substrate. But notice how the enzyme is unchanged. And if this is really confusing, the image here makes it a lot easier to understand. So we have our substrate. It's going to bind with our active site of our enzyme. When they come into contact, we form our enzyme substrate complex, and we form our products. We broke down the substrate in this example, and now we're joining the substrate together in this example. And notice how this active site is going to look a little bit like the substrate here. They have to kind of fit together. Here's a great animation of what I'm talking about. We have our enzyme, we have our substrate, it binds together, it forms that enzyme substrate complex, they break apart, and our enzyme is unchanged, it's reusable. I'm not going to show this video, it's a good video, but I'll come back to it. Specificity, this is the idea that enzymes are specific to one type of molecule. They are specific like a lock and a key. So only certain or a small amount or just one substrate will fit within a enzyme. Again, this is based on the structure of the enzyme and the substrate. So you can see here different enzymes, different substrates, they all are specific. Good meme. Yeah, they have to be specific to their enzyme. All right, last slide here, induced fit. So what induced fit is the concept that I just told you that the enzyme and the active site match the substrate. It's not perfect. You can see here our enzyme looks a little bit like our substrate. What happens is they'll kind of form around each other, and with that formation, that kind of change in conformation, that's where the reaction then will take place. This is called the induced fit model, and this is a really good animation of what that looks like. So the enzyme looks like I don't know why it's not playing. Yeah, I can go back to actually this slide. The enzyme looks like the substrate a little bit. Here we go, but it's not going to be perfect. I don't know why it's not playing. I'll have to fix it on my PowerPoint. But this is called the induced fit model, and this is the end of 6.2. Oh wait, no, it's not the end of 6.2. 6.2. All right. So the energy of activation. This is again that we talked about how the reactants form the products. What enzymes do is they act like a catalyst, and they act like a catalyst in the sense of they are going to lower activation energy. We talked about how we have reactants, products losing energy. In this case, it lowers the activation energy for a reaction to take place. So again, this increases the speed of the reaction. And how I like to remember this is we have this kind of barrier from the reactants to our products. We have our frogs here. They're trying to jump over to become the products. They have this activation energy. Enzymes lower that activation energy. So you can see without enzymes and then with enzymes. Our next chapter, our next section is going to be on 6.3 factors that affect reaction rates. So we're talking about enzymes and how they react with different substrates. And we know enzymes can do one of two things. They can build things up or they can break things down. One of the factors that affects how fast an enzyme can react is called substrate concentration. So as you know, substrates go into reactions with enzymes. The more substrates we have, the more reactions that can occur until a certain point. This point is called the point of saturation. Now, this is kind of hard to understand. And what I would do is I would draw this down in your notes. I would draw this graph of having this point of saturation. What this looks like, and I know it's a little bit fuzzy here, but as we add in more substrate, you can see there's going to be more reaction. This is just because there is just more of the substrate for the enzyme to react with until a certain point. Again, this is the point of saturation where it doesn't matter if we keep on adding in more and more substrate, it's going to saturate. It's going to be a point where it doesn't matter how many substrate or how much substrate is in there. You have enough substrate for all the enzymes to be working at all times. Temperature is our next one. So temperature influences reaction rates. This is the graph here. You can see here we have our enzyme rate, we have our temperature, we have our optimal temperature right here. As we increase temperature, you're also going to increase the rate of reaction until a certain point. Now, what happens is after this certain point, the protein becomes denatured. We talked about that a little bit back in chapter 3. We're going to come back to that concept here in this chapter. And that's because when you add in heat, you're adding in energy, and you can see what's happening here. As we add in more and more energy, these molecules are going to keep on bumping around until when we have a lot of energy, they're really just bumping around. So they're going to come in contact with that the active side of the enzyme more than over here. You can see there's not a lot of energy here, more energy here, and a lot of energy here. And the last one I like to talk about is the optimal pH of enzymes. Again, this is very similar to the temperature. However, this is a little bit different. You can see this graph it's kind of the same on both sides. Instead of having it going slowly up as in temperature, it's even on both sides. And again, over on this side and over on this side, the enzyme's also going to be denatured. So this is being denatured, this is being denatured. Remember what denature means. We're going to go over it here in a little bit, but you're unraveling the protein. The structure of the protein is going to be affected, so you're unraveling that protein. And this is just a graph to show you that enzymes have different optimal pHs. So some will have a pH around 7, some will have it around 2, some will have it around 10. It just depends on where that enzyme is going to be found. Now, denaturing. This occurs when the protein starts to break down due to things like temperature and pH. You can see what denaturation looks like. The protein is going to lose its shape, which means it's going to lose its function. Now, you can renature the protein, yes, but we don't really focus on that. We typically look more on denaturation and how the protein denatures. I'm going to go back a couple slides. So again, if you put a protein in let's say this protein in a solution of 11, it's not going to function properly because it's going to be denatured. If you put a protein, an enzyme, in a temperature that's over on this side, it's not going to function properly because it's denatured. The structure is affected, which means it affects the function. All right. Good. We're going to keep on pushing forward because I just want to go over the important stuff here. All right. That's actually the end of 6.3. Yep. End of 6.3. Our next section in this chapter is going to be on 6.4 inhibitors. Whenever you have an inhibitor with an enzyme, it's going to stop that enzyme from occurring or from working with the substrate. The first type of inhibition is going to be called competitive inhibition. If you think about competing, you're thinking about competing for the same site as the substrate. So in this enzyme reaction, we have our substrate meeting the enzyme, it gets broken down. A competitive inhibitor is trying to compete with the active site for that substrate. So that substrate is not going to be then able to bind with the enzyme. I think of this as like if you have a lock and a key, if somebody puts a different key in the lock and it gets stuck, it's competing for that same area, that same active site of that lock. So again, it's competing for the active site of the reaction. I like showing this animation here. You can see the competitive blocker here. It's binding with the active site, which means the substrate can't bind with it. There you go, and no reaction occurs. Good meme. There's a video here I'm not going to show you. Non-competitive inhibitors. This is where the inhibitor will bind to a location not on the active site. It's going to be away from the active site. Now, what that site is called, it's called the allosteric site. This causes the active site to change shape, which when you change shape, you change function. So you can see in this reaction, our enzyme would normally bond with the substrate, but the inhibitor is changing the shape, which means there's no reaction. Again, other videos I'm not going to show. We can see our non-competitive blocker here or inhibitor. It's changing the active site's shape, which means it's not going to be able to bind with the substrate. And that stops the reaction. Now, one more thing I like to add in here is, good memes here, there is an allosteric activator. So when this happens, it's kind of the complete opposite, where this enzyme won't function unless there's an activator that changes the shape. So instead of inhibiting the enzyme, this can actually activate the enzyme. And this is kind of what we talked about with kinases. The kinases will activate certain proteins by giving them a phosphate group. So again, I'll show this quick video.
Allosteric regulators.
Maybe not. Okay. This is the end of 6.4. I think it's the end of 6.4. Yeah, 6.4. All right. The next section is going to be on something called compartmentalization. This is a pretty significant concept to understand for this chapter. So really pay attention to this chapter because it's really going to apply not only to this chapter, but to other chapters and other units. So in compartmentalization, we have something called a metabolic pathway, where this is a series of linked reactions. Think metabolic, all your reactions going inside your body. Pathway, just a series of events. So we have our substrates and products are these molecules A, B, C, D, all the way to G. Every one of these E's represents an enzyme. So A is the substrate for E, which makes B. B then is the substrate for E2, which makes C. C is the substrate for E3, which makes D. So you can see the substrate for each enzyme is the last enzyme's product. This is what we call an enzyme pathway or a metabolic pathway. You can see again, A goes into E, makes B, B goes into E, makes C, so on and so forth. This is an enzyme pathway. I like showing this animation. You can see the enzyme pathway at the top there. Each one of these substrates are going to come together in the enzymes and finally form the product at the end, but they have individual steps in between. Here's another way of looking at it. You can see each of these enzymes here is going to change this tyrosine into melanin. The difference between an enzyme pathway and an enzyme cycle is there's really no endpoint for this cycle. You can see it's going to go around and around. This is going to set us up for the next chapter, which is going to be on photosynthesis and cellular respiration. Now, feedback inhibition. This is when we have this enzyme pathway, and the end product here is going to inhibit one of the earlier steps. We talked about these inhibitors a little bit earlier on, I think in 6.4. What you're going to see here is this end product stops this enzyme from working. That subsequently stops everything else from occurring in this process. Why do you think this occurs? The answer is efficiency. If you already have enough of this product, why make more of this product? We don't need to make more. We already have enough. It's not efficient. So what happens is this goes back, stops this process. It's efficient. Now, we have things called metabolic pathway diseases. Metabolic pathway diseases are when a mutation occurs and for some reason an enzyme doesn't work, which means we get an overexpression of E, and then we get less or no F and G in this cycle. So there's two problems here. The end product isn't made, and then we have this overabundance of E. And we're going to see this metabolic pathway later on in the year, but this can cause major problems within people. And here's a couple different examples of the enzyme pathway diseases, where if one stops, you can see which is built up, and then again, none of them we have the product being formed. All right, 6.5. We're just going to go on to 6.5. This is 6.5. I want to stop it before 6.6. Compartmentalization. So what does this allow us to do? Compartmentalization is when smaller spaces have more significant purposes, like functions. I say purposes, but functions. This allows the organelles of a cell to work more efficiently. If we put all of the things inside of an organelle that we need to occur, like say for cellular respiration or photosynthesis or the rough and smooth ER or a vacuole or a Golgi body, we make it easier and more efficient for the cell to go through that process. And this is because we're raising substrate levels and we're raising the collisions before with all the molecules and their substrates. So again, we're decreasing. I'm going to bring my video up here. We're decreasing the surface area to increase the substrate and enzymes bumping into each other. That allows the cell to really increase the efficiency of that particular process. And that's the whole kind of concept, right? Our last section in this chapter is going to be on 6.6, which is redox reactions. This is the transfer of electrons from one molecule to the next. We also call them redox because reduction, oxidation reactions. If you're like, oh my gosh, I remember these from chemistry, it's not that bad. All you have to remember is where the electrons are going. So in an oxidation reaction, there's a loss of electrons. In a reduction reaction, there's a gain of electrons. It sounds kind of opposite. You think when you reduce something, you lose something. But in this case, we're kind of looking at the positive and negatives of things. So don't go too far into it. Just remember oxidation is losing, reduction is gaining. I'll show you how to remember that here in a second. But again, molecule A is being oxidized because it's losing these two electrons. Molecule B is reducing because it's gaining those electrons. This is how I remember it. Oil rig. Oxidation is losing, reduction is gaining. So remember oil rig to remember oxidation is losing, reduction is gaining electrons. I'm just going to preface this. We're going to see a lot of these oxidation reduction reactions within photosynthesis and aerobic respiration. So that's like kind of why we're hitting these enzyme pathways, and then we're hitting the enzyme cycles and these redox reactions right before the next two chapters, because this is going to be really important. But this is the end of chapter 7.